Hi everybody, I am Dr. D.S. Thore. Now I share the screen of today's lecture in front of you. Please wait. Okay. Welcome to the second lecture of course number PATH-365, that is the diseases of field and horticultural crops and their management. In today's lecture, we will discuss about downy mildew disease of field and horticultural crops and also vegetable crops. In last lectures, we already discussed the syllabus of this course. So in today's, we discuss about the downy mildew disease seen in field crops, horticultural crops, and also vegetable crops. Okay, so this is the number of the pathogens responsible to produce the such disease in fruit crops, also field crops, and also the vegetable crops. Okay. For example, downy mildew disease of grapes caused by Plasmopara viticola. Downy mildew disease of sunflower caused by Plasmopara halstedi. Downy mildew disease of onion, Perinospora destructor. Downy mildew disease of mustard caused by Perinospora parasitica. Downy mildew disease of cucurbits, Pseudoperinospora pubins. And last one, downy mildew disease of pea caused by Perinospora pisci or Perinospora vicia. Okay, so this is the number of genera causing the downy mildew disease in particularly grapes, sunflower, onion, mustard, cucurbits, and pea. Okay, so just to see one by one, that is the first one, downy mildew disease of grapes. Okay, so this Plasmopara viticola pathogen infect to the grapevines and produce the number of the symptoms on the leaf, shoots, branches, then growing bunches. Means this symptom also be developed on the fruiting stage of the grapevines. Okay, so you just to see this number of the spots generally seen on the upper side of the leaf. Okay, so basic symptoms generally produced by any genera of the pathogen, that is symptom generally develop on the both side of the leaf, means corresponding upper side of the infected leaf showing these chlorotic patches. Okay, means initially the small yellow translucent spots generally scattered or seen on this infected leaf. Okay, so these chlorotic spots just enlarges over this infected leaf and showing the large blight or just the largest chlorotic spots or blight lesions may be developed on the infected leaf. Okay, so this type of the symptom generally seen on the upper side of the grapevines. Okay, okay. Then these pathogens just grows inner side and develop the symptoms on the lower side of this same leaf. Okay, means on the underside of the leaf, the white downy growth of the fungus are seen. Okay, means these infected areas showing the maybe reddish line in colors. Means initially just the whitish downy growth generally seen on the lower side of the leaf. So under humid conditions, this downy profuse growth is spread to the entire surface of the leaf. Okay, means in the favorable conditions, this downy profuse growth may be spread or may be seen on the leaf, then shoots, then tendril parts of the grapevines. Okay, means finally this downy growth also be seen on the flowers and developing the berries. Okay, means during the blossom or early fruiting stages, the clusters or some parts of the clusters of the berries develop this whitish downy growth. Okay, means the infection takes place after the formation of the berries. So this fungus grows mostly internal parts of the berries. Means these infected berries become leathery and wrinkles. So somewhat these reddish to brownish discolorations generally seen from these infected berries. Okay, means such infected berries is said means retard the quality and quantity of the berries, in particularly bunch. Means the juice quality of these affected berries is reduced. Okay, means somewhat these infected tissues turns brown and dies. Okay, means this Plasmopara viticola pathogen infect to the leaf, infect to the shoots, infect to these tendril portions, and infect to the berries, in particularly bunch. Okay, so this is the major symptom generally developed on the different parts of the grapevines due to the Plasmopara viticola. Okay, then Plasmopara viticola pathogens develop the mycelia, and this mycelia is intercellular, and somewhat this mycelia grows into the infected host tissue by forming the hostillia. Means this mycelia is generally sinusoid, hyaline, and thick wall. Okay, so just to you see this life cycle of this Plasmopara viticola. Means this is the mycelia grows intercellularly in between the host tissues. So under favorable conditions, this mycelia grows and to produce the erect sporangiophore. Okay, so this sporangiophore is 300 to 500 micrometer in long and somewhat 7 to 9 micrometer in wide. This size of the sporangiophore is near about 300 to 500 by 7 to 9 micron in diameters. Okay, then this sporangiophore is branched, is almost right angle to the main axis. This is the main characteristics of the Plasmopara viticola. Means these Plasmopara viticola pathogens develop the branching structure right angle to the this main axis. Okay, so on the basis of branching structure of sporangiophore, you can properly identify the particularly genera of the downy mildew. Means in case of the Plasmopara viticola, the branching structure is generally takes place at the right angle to the main axis. Okay, so at the tip of the branching structures, somewhat 2 to 3 stigmata is developed. So this is the 1, 2, and 3. Means at the end of the stigmata, the oval set sporangia generally attached. Okay, means this sporangia is a thin-walled, oval, and lemon cell. Okay, so this sporangia is germinating and finally develop the juice pores. Okay, means biflagellate juice pores is produced from this sporangio, means sporangium. Okay, sporangium, it is the fruiting body of the fungus developed by asexually manner. Okay, so this sporangioscore or sporangia and juice pores disseminated from this infected host tissue and deposit on this healthy one host tissue. So these sporangia and juice pores generally fallen on this healthy one host tissue. So these spores generally grows intercellularly in between the host tissue. So this host tissue become infected by forming the hysteria, means finally develop the disease in the healthy one host tissue. Means this healthy one host tissue become infected. Okay, means this Plasmoptera viticola pathogen reproduce through asexual method by forming the zoospore and sporangia. Okay, so this is the spores developed through asexually methods. Okay, then the Plasmoptera viticola pathogen reproduced by sexually methods means the zoospores germinate under the favorable conditions and develop the disease on that grape trees. Okay, means these oospores developed by male and female gametes fuse to each other. Means suppose this is the oogonium and this is the antheridium. Means male nuclei migrate toward the female nuclei. So finally develop the oospores. Means oospore is a spores produced by sexually reproduction methods. Okay, means two-walled sexual spores is produced by Plasmoptera viticola. Okay, means in this case, perpetuations generally takes place through oospores and zoospores. Means in this case, primary infection takes place through infected leaf or infected any parts of the grapevines, generally deposit or decrease in the soils. So finally, this oospores is produced. Okay, means oospore present in soil or infected plant part decrease in the soils. So these two sources acts as a primary source of infection. Okay, but secondary source of infection generally takes place through these sporangia and zoospores disseminated from infected host tissue to the healthy one host tissue through windborne or means any wind agency or through water or moisture present on the infected host tissue. Okay, means in short, oospores and infected plant parts acts as a primary source and zoospores and sporangia acts as a secondary source of infection seen in Plasmopara viticola. Okay. Then management. Okay. So first effective management, that is removing and burning up all the infected leaves, shoots, flowers, and berries. Okay. So this is the source for hibernating the or survival the ooze spores in the absence of the host plants. Okay. So it is necessary to collect such infected leaf or any plant parts and burn properly. Then after pruning, this grape wine should be sprayed with the borax mixture 1% or dipolydol 0.2% or chlorothalonil 0.2%. Okay. Then grape wine field should be kept high above the ground to allow the circulation of the air by proper spacing, because relative humidity is a major factor to produce the downy growth on the grapevines. Okay, so it is kept, this grapevine field is free from disease to allowing the air circulation properly in particularly grapevines. Okay, suppose this downy growth is seen on the any parts of the grape trees. So immediately you can apply the 1% borax mixture or phosphatyl AL, that is allied, or 0.2% phosphatyl AL, okay, or metalaxyl plus mancozeb, means redomil. This is the redomil fungicide is the best effective to control against these downy mildew disease in grapes by using the 0.2 or 0.3 to 0.4%. Or recently, these azoxystrobin, this fungicide is also used for to controlling this disease. Okay, means use of the borax mixture as a 1% on grape trees immediately after the pruning of the vines. This is the first stage. Then second stage, new flowers should form. Then third stage should be done at the buds opening. Then fourth one, bunches or berries have been formed or during the growth of the shoots, because this downy mildew fungus infect to such grapevines in the various growth stage of this fungus. Sorry, this various growth stage of the grape. Okay. Then also use the biological control measures. For example, Irvinia herbicola. This is the bacteria is also used to control this Plasmopara viticola disease in the grapevines. Okay. Then second most important disease seen in field crops, that is downy mildew disease of the sunflowers. Okay. So this is the some photographs of the sunflower causing the Plasmopara history, means finally produce the downy mildew disease of the sunflowers. Okay, means this fungus infect to the seedling stage of the sunflower, means early growth stage of the sunflower, as well as the growing sunflower plants. Okay, so this fungus infect to various growth stage of the sunflowers and produce the number of the symptoms, that is damping of symptoms, then foliar lesions, or sometimes these whitish downy growth seen on the lower side of the leaf, that is known as the systemic infections, or in the heavily inoculum load of these pathogens. So basal root or stem gall symptoms also be seen on the infected stem and root surface of the sunflowers. Okay, so just we see this first symptom generally develop on the growing or just engaged plants of the sunflowers, because this primary infection takes place through the wood spores present in the soil. Means these wood spores directly infect the growing seeds of the sunflowers. Means these seeds germinate and to produce the seedlings. So these seedlings may be uh, means these seedlings get infected by the fungus and showing the damping of symptoms. Means in this stage, the seedlings are killed before or soon after the emergence of the seeds. Okay, means this infection generally seen on the growing younger seedlings of the sunflower. So such infected seedlings not properly developed means retard the height of the plants and develop the damping of symptoms. Okay. Then the second most important symptom generally seen on the leaf portions means on the growing sunflower plants, the initially yellow chlorotic lesions is generally develop on the upper side of the leaf. Then this fungus also be develop the symptoms on the lower side, means the whitish profuse growth generally develop on the lower side. So this under favorable conditions, this whitish profuse growth is spread to the entire surface of the leaf. Okay, means under favorable conditions, this downy profuse growth spread from leaf to the stem portion. Okay, and other growing parts of the sunflower plants. Means this fungus become systemic in nature. Okay, means this fungus generally develop on the flowers as well as the head portion of the sunflowers. Okay, means at the time of the flowering seasons, this whitish powdery growth may be seen on the flower. So these infected flowers is become chlorotic and just dries up. Okay, or this uh, whitish growth also be seen on these head portions. Okay, so these flower heads of infected plants remain erect and also small in size as compared to the normal plants. Okay, means these flower heads may or may not be sterile in nature. Means sometimes sterile or sometimes only few grains is formed in this infected flower heads. Okay, means grains is formed or no grain is formed within these infected flower heads. Okay, so these infected plants is totally stunted in nature. Okay, and last symptoms means this symptom generally develop on the stem portion or root portion under heavily infection seen on the ground sunflower plants. That is sometimes stem galls seen on the stem portion or root portions. Okay, means these galls is developed at the base of the plants. Okay, may be developed on the stem or may be developed on the root portions. So such infected plants are less vigorous and generally these infected plants is losing takes place because this infection majorly seen on the basal side of the or basal portion of the sunflower plant. So such infected plants become lodged. Okay, so this is the symptom generally developed on the sunflower plants at the seedling stage as well as the growing stage of the sunflower. Okay, so this is the majorly symptoms seen on the leaf portion as well as the head portion or flower portion of the sunflowers. Okay, so this is the downy growth generally takes place on these flower heads. Okay, so these flower heads is remain sterile in nature. No any grains are formed in such infected flower heads. Okay. Then Plasmoptera halsteadi. So this pathogen develop the mycelia, and this mycelia is non-septate, sinuositic, and produces the hysteria into the infected host tissue. So finally develop the sporangiophore. So this sporangiophore generally branched at the right angle to the main axis also. Already I explained these characteristics of the Plasmoptera viticora. Okay, means sporangiophore generally branched at the right angle to the main axis. This is the main characteristics of the Plasmoptera genera. So same case generally seen in also the downy mildew disease of the sunflowers. Okay, other primary source and secondary source is same. That is the primary source means infected seeds or ooze spores present in the soil or infected plant part debris in the soil. So this is the mainly source for initiate the disease in the sunflower fields. After the infection of the pathogen to the sunflower plants, so these sporangia and zoospores generally develop, and these sporangia and zoospores liberated from infected host tissue, the other host tissue, means healthy one host tissue through wind or water. So in this way, spread the disease from infected plant to the healthy one plants. So in this way, spread the disease from one place to the another place on particularly sunflower fields. Okay. Then management. So this is the management practices done by the every farmers to avoid the downy mildew infection in sunflower. Already I told these management practices in grapes. That is sanitation programs to be run to avoid the primary source of infections. Then follow spacing that is a 60 by 30 centimeter or 45 by 30 centimeter. Then rub out the infected plants and destroy properly. Rub out means locate the individual infected plants and uproot such a plant from this field. Then cropping means crop rotation of the sunflower by adopting the groundnuts also. Okay. Then seed treatment, because this disease is also initiated through use of the infected seeds. So these seeds is a disease-free seeds by applying the fungicides. We just take the metalaxyl powder, and this metalaxyl powder is putted over these sunflower seeds. So in this way, you can protect the seeds from the fungal infections. Okay, then foliar sprays is also necessary to avoid this disease in the sunflower. That is Ridomil, MZ at the rate of 0.2%. This fungicide is effective on 20 days after the sowing, because this infection seen on the early growth stage of the plant, means at the seedling stage of this groundnut, this sunflowers. Then growing some cultivars or resistant varieties, that is LSH1, LSH3, KBSH1, Jalmukhi. So this is the number of the resistant variety grown by the farmers to avoid such type of the infection in the sunflower plants. Okay. Then third most important disease seen in also onion. Okay. So in this case, the systemic infections generally occurs when the plants are growing from the diseased bulbs. Okay, means in the favorable conditions, the pathogen infect to this growing leaf of these onions. Means initially the whitish chlorotic lesion seen on this growing leaf of the onions. Okay, so this infection is spreaded deeply to this tissue of the onions. So under favorable conditions, this whitish downy profuse growth is seen on the leaf surface. Okay, means in the humid conditions, the whitish downy growth are seen. But in the dry conditions, somewhat only white dots are seen on the surface of the leaf. Okay, means the this chlorotic zones interrupt with the greenish zones. So such type of the infection is seen on the leaf of the onions. Okay, means usually this infection generally seen on the lower side of the plants. Means older leaf get first infect and showing the chlorotic and whitish dots are seen. Okay, so under favorable condition, this disease is also spread in or from this leaf to the downward directions. Means uh, these pathogens spread up to the development of the bulbs. So these bulbs produced on the deceived plants can remain small with a succulent neck. Okay, so succulent neck are subject by attack by the number of the fungi in storage conditions. Okay, so this is the whitish profuse growth is seen on the surface of the leaf. Okay, so this is the necrotic lesions or somewhat these burning leaf generally seen on this onion field due to the Perinospora destructor pathogens. So this is the heavily infested plot of the onion due to the Perinospora destructor. Okay, so this is the somewhat these number of the fungi may be develop the symptoms on the developing the bulbs. In such bulbs is always remain small in size as compared to the healthy one plant of the onions. And this neck portion is succulent. For a number of the fungi and bacteria also invades such succulent leaf. Okay, then ecological parts of this Perinospora destructor. Okay, so other morphological character is same, only differ is the sporangiophore. Okay, means in this case, this sporangiophore is a non-septate, means no any cross wall or septa is present on this growing sporangiophore from this mycelium. Okay, so this branching of this sporangiophore it generally takes place on the top portion of this sporangiophore. Means this sporangiophore is remain unbranched up to 3/4 portion of this sporangiophore. Means always a branching structure is takes place on the top portion of the sporangiophore. Okay, so this sporangia is a pyriform or fusiform and attached to this stigmata. Okay, so this sporangia germinate under the favorable conditions and to produce the biflagellate juice spores. Okay, means in this case, secondary infection takes place through sporangia and juice spores is disseminated from one place to the another place through wind or air currents and also the moisture means with the help of the moisture. Okay, Okay, so this is the sporangia is produced or attached at the tip of this stigmata. Means branching structure is seen at the top portion of such a sporangiophore. Okay, so this is the disease cycle of the Paranospora destructor. Means in this case, oospores is formed by fission of the male and female gametes. So finally, oospores is germinating under favorable conditions and to produce or attack to growing onion plants. So these onion plants become infect, means finally develop the whitish mass seen on the leaf portion. Means already sporangia and zoospores is formed, and these zoospores disseminated from this infected host tissue to the deposit on the healthy one host tissue. So this healthy one host tissue become also infected. So in this way, primary infection and secondary infection is expressed. Okay, then management is same in onions. That is a seed treatment with metalaxyl or thiron or carbamazine plus thiron. Then field sanitation, then spraying with the metalaxyl. So instead of metalaxyl, you can also apply the mancozeb or use the Xyron or dichloride. Okay, so spraying should be started 20 days after the transplanting and repeated the number of the sprays at 10 to 12 days intervals. Okay, then next important disease that is a downy mildew disease also seen on the mustards. Okay, so in case of the mustard, the basic symptom that is the yellow chlorotic spots generally seen on the leaf of the mustards and the corresponding lower side of the such a leaf having the whitish profuse growth are seen. Okay, is under moist or cool conditions, this leaf spot enlarges indefinitely to form the yellow chlorotic areas on the upper side and lower side, that is a whitish downy growth seen on the lower side. Okay, means this fungus becomes systemic in nature. Okay, means the tissue of the infected leaf become dries out and abscission layer means this infected leaf is mainly separate from this stem of the mustard plants. Okay, means the infected leaf generally dries and separated from the mustard plants. Suppose this infection seen at the flowering or any reproductive organs of these mustards. So these infections seen on inflorescence means hypertrophy symptoms is developed. Means these reproductive organ is totally sterile in nature because this downy growth is seen on the any reproductive organ. So these reproductive organs is not properly developed. So that's why these reproductive organs is hypertrophed in nature. Means no any pods are developed instead of the flowers. Okay. Then causal organism that is a Perinospora parasitic. Okay. So this Perinospora means all the downy mildew pathogen are obligate parasite in nature. Means obligate parasite means this parasite cannot survive without the host plant. Means always survival with the help of the host plants. Okay, so this sporangiophore generally branched at dichotomously means only two stigmata is developed from the single branch of this sporangiophore. Means sporangiophore is dichotomously branching. Okay, dichotomously means only two stigmata is developed on single branching structure of this sporangiophyll. Okay, so this is the basic criteria to identify the Perinospora genera. But in regarding the Plasmoptera genera, two or more than two stigmata is developed on the branching structure of this sporangia. Okay, so this sporangia generally attached to this stigmata. Okay, means in this case, also primary infection takes place through wood spore present in the soil or infected plant part debris in the soil or contaminated seeds is used for sowing purpose. So 100% this is produced. Okay, and secondary infection takes place through the sporangia and juice spores through water or wind bonds. Okay, so this Downy mildew disease having the wide host range because this Perinospora parasitica pathogen also develop on the cabbage, cauliflower, novel pole, radish instead of the mustards. Okay. Then last one that is control measures means well drained and cleaned soils generally used for or cultivation of these mustards or avoid the cultivation of the any cruciferous crops near to the field of the mustards because these Perinospora pathogens having the wide host range. Then crop rotation with non-host cruciferous crops. Then before the actual sowing of the mustard seeds in the main field, so these seeds is treated by applying the hot waters. That is just to take the number of the seeds and uh, dipped in the hot water having the temperature that is 50 to 52 degrees Celsius for only 1 or 2 minutes. After that, this seeds is set in dry conditions and immediately used for sowing purpose. Or this seeds is again treated with the Captain Thyron or any acid treating fungicides and immediately used for sowing purpose. Then you can apply the Borax mixture 1% or Manab or Zenab 0.2% at 10 or 15 days interval to check the disease properly or spraying that is a copper oxychloride 0.3% starting at seedling stage of these mustard plants. So subsequently you can apply the 3 to 4 sprays by using the any fungicide that is a copper oxychloride or manganate or mancozeb at an interval of the 10 days because this is the means this disease is mainly inhibited by applying such fungicides. Okay, then downy mildew of cucurbits. Okay, so in this case, the basic symptom is same that is on the surface of the uh, leaf is on the upper side just fluoratic patches is seen. So these fluoratic patches is irregularly spread to entire surface of the leaf means upper side of the leaf become fluoratic and showing the yellow in colors. Okay. Then under favorable conditions, this whitish profuse growth is seen on the lower side of the leaf. Okay. So you can see this number of the symptoms, that is the chlorotic patches or spots generally seen on the leaf. So this infected leaf generally dries, and such a dry leaf is separated from this cucurbits vines. Okay, then causal organism, that is the Pseudoperinospora pubescens. So this is the obligate parasites. Then the main characteristics of this Pseudoperinospora, that is the upper third portion of the sporangiopore is branched either dichotomously or intermediately between the dichotomously and monocotomously branching. Means one stigmata or two stigmata is developed at the end of the branching structure of the sporangiophore and generally attach the sporangia. Okay, this is the main characteristics of the Pseudoperinospora pubescens. Okay, so you see this branching structure of the sporangiophore means just one or two stigmata is developed on this branching structure of the sporangiophore. So at the tip of this stigmata, generally sporangia generally attached. Okay, so you just see the sporangia spore with the sporangia. Okay, so this is the life cycle or disease cycle of the Pseudoperinospora pubescens. Means this is the whitish downy root generally seen on the lower side of the leaf. So this means this is the underdevelopment of the pathogen by forming the posterior and to develop the sporangiophore. So at the tip of the sporangiophore, stigmata is developed. This finally sporangia is attached to this stigmata. So this sporangia liberated from this sporangiophore means this infected plants and deposit on the healthy one host tissue. So this healthy one host tissue also infected by forming the posterior. Okay. Primary infection is the same, that is the root spores that infect the plant particles in the soil, and secondary infection, that is airborne sporangia and juice spores or moisture generally seen on the surface of the cucurbitaceous plants. Then management is the same management points. Okay, that is a sanitation program. Then avoid the successful planting in adjacent field during the single season, reduce the spread of the from early planting to late planting. Then chemical fungicides generally spread over the host plants. Then seed treatment. Okay, so this is the same management practices adopted by every farmers to avoid the downy mildew infection in the cucurbitaceous plant. And last one, that is downy mildew disease of the pea. Okay, so this is the major disease and most devastating disease seen in the pea plant also. Means in this case, the infection of the pathogen starts as a small chlorotic spots seen on the lower side of the leaf when the plants just 4 to 5 leaf stage. This infection generally seen on the seedling stage of the pea. Okay, so this whitish downy growth seen on this engaged leaf. So these younger plants infect and become wilted in appearance. Okay, means this upper leaf surface of the tree infect with the downy growth. So this downy growth become greenish yellow to brown blotches seen on the number of the leaf of the infected pea plants. Okay, so this yellow chlorotic patches seen on the upper side of the pea leaf. Okay, so this downy growth Means a whitish downy growth seen on the older leaf or older branches of the pea plants. Okay, means in this case, suppose this whitish downy growth seen on the younger seedlings. So this stem is also infected and leading to the systemic infection on the growing plants. In this infection seen on the development or at the time of the development of the pods. So these pale green whitish lesions also develop on the developing pods of the pea. So these pods is totally white in color, having the grain is also small in size or chappy in appearance. Okay, means this small size is produced in or within this infected pods seen on the infected pea plants due to the downy mildew pathogens. Okay, then causal organism that is a Perinospora PC. Okay, so this mycelia is a sinuosity and develop this sporangiophore. So these sporangiophore are unbranched in the lower two-third portions, and this remaining one-third portion is a 2 to 10 times dichotomously branching. Means this sporangiophore is unbranched up to 3/4 portion of the sporangiophore. Means a sporangia is attached at the stigmata. Okay, so other ecological parts is same. So you see this life cycle of this Perinospora PC. Okay, so this infected leaf having the yellow chlorotic patches or maybe whitish downy growth are seen. So such infected plants or infected leaves just debris in the soil. So these infected plants having the wood spores is survival for several years under horrible conditions. These wood spores infect to these younger plants of the pea and develop the symptoms on the leaf. Or suppose this seed is collected from these infected plants and used for sowing purpose. So 100% this is the produce. Okay, means already this sporangia is disseminated from this infected host tissue and just fallen or deposit on the healthy one host tissue. So this healthy one host tissue become infected. Means in this way spread the disease from one plant to the another plant through this sporangia and juice spores through wind or with the help of the moisture. And primary infection takes place through these infected seeds and infected plant parts or maybe wood spores survival in soil up to few years. Okay, then control measures, field sanitation, destruction of the infected crop debris, 3 to 4 years crop rotation with non-host crop. So this is the cultural practices is important in reducing the primary inoculum. Then always use the disease seeds, then seed treatment with hot water having the temperature 50 degrees Celsius for 24 minutes. So this seed treatment has been found effective to eradicate the seed-borne inoculation. And the last one, spraying the crop with metalaxyl at the rate of 0.2% or chlorothalonil at the rate of 0.3% or Curzet at the rate of 0.12%. So this fungicide is applied at an interval of 15 days. So you can avoid or prevent this downy mildew pathogen on the pea plants. Okay, so in today's lecture, I discussed about the downy mildew disease seen in field crop, horticultural crops, and vegetable crops. Okay, so keep in your mind the basic symptoms seen on the upper side, that is the chlorotic patches seen on the upper side, corresponding lower side, downy growth are seen. This is the basic symptoms. Okay, then how to identify the proper genera of the downy mildew disease or downy mildew pathogens on what basis? So this branching structure of the sporangiospore. So this is the basic principle or criteria to identify the proper genera of the downy mildew pathogens. Then primary source and secondary source. That is, the primary source means infected plant part deposits in soil. That is second source, infected plant part deposits in the soil, and third source, that is an infected seed used for planting purpose. So this is the three main source to initiate the disease in the host crops. And secondary infection is always takes place through the sporangia and zoospore, spread through wind or air current, and moisture is also role for flow these zoospores from one place to the another place. And management, that is a sanitation program is common. Then second, avoid the non-host crop, because this Peronospora or other genera, genera of the pathogens having the wide host range, means avoid the such host crop with the respective crops, means crop rotation with the other non-host crop. Then collect and destruction of the affected plant parts. Okay, then seed treatment with metalaxyl or any captain therum, etc. And last one, foliar sprays with the metalaxyl or mancozeb or other borax mixture, copper oxychloride. This is the mainly fungicides is generally applied over this particular crop for to check the secondary source of infections. Okay, so this is the summary of today's lectures. Okay, thank you.